how is this related to CRISPR or gene editing, like protein visualization, if anybody could add. Yes, exactly. We can check that. In mutations where the stretch of the mutation is like where there is deletion mutations, like a long stretch of your target gene is mutated and deleted, then in that case your protein will not be translated. So, but if you have some insertions or deletions and there are some frame shift mutations, then that will affect the protein expression levels and the structure of the protein as well. So that's why I'm discussing this in this tutorial session. So I can remember that Aina told you about that Swiss model where you can build your own model, and she didn't, I think she didn't tell you about the protein, the 3D structure visualization tools. Okay. So here first we'll take an example of any protein that you can think. Like I have just searched, you can just open NCBI, and here you can in this drop down section you can just select protein, so that it will give you only the protein, result of only the protein. So it's giving me the result of this 16-dox, which is also called this 2-oxoglutarate dependent dioxygenase in case of Solanum lycopersicum. So I'll be clicking on that. And after clicking on that, you can simply just 16-dox. You can search 16-dox or any other protein, but you have to have that. Okay, you can search any protein for now. Protein, yes. So it will give you only the result for protein. And in the previous tutorials, I have told you how to retrieve this sequence. You have to click on that protein part. Yes, and after that you will be able to come here. Okay. So after clicking on this Solanum lycopersicum 1, you can just scroll down and select this protein, click on that protein, and at the extreme right bottom corner you can just click on the FASTA, and it will give you a FASTA sequence for this protein. Okay, so have you all opened NCBI? You just have to open NCBI first. Let me just open it in a different tab. So you will just open NCBI, and from this drop down menu you can select protein, so that it will only give you protein result. So after that you can type 16-dox or any other protein that you want to visualize. So after typing it, you can just search. And after that, are you following along? Okay. So after that you will click on that, and in the bottom you can find this protein written over here. So you will just click on it and it will highlight this whole sequence, and you can just click on the FASTA written over there at the extreme bottom, and after clicking on that you will be able to get the sequence, protein sequence for this. So you can select it as I have selected it and copy it. Have you all copied it? Yes. So you can open Swiss model. Now you all must be familiar with Swiss model, so you can just open the page and click on Start Modeling, and you can simply paste your selected protein sequence here and search for templates. And why is this? Okay. Let it search for the templates, and after that we'll select one template, and based on that template we'll build a model. And meanwhile, there are several tools which we can use for visualization, protein 3D structure visualization. One of that is UCSF Chimera. You can just type it on your Google browser, UCSF Chimera, CHIMERA, and you can search for it. And there you will be able to okay, so this is the home page, and you can see in the left this download icon. You can just click it and download it according to your laptop, whether it's running on Windows or on Mac. It's available for all the platforms. Yes, this is the link. You can just see the cursor here. So it's similar like how you install any other program. So you can just install it. As I have already installed, so I won't be installing it again. So for Mac users, you can just click on the link here. Okay. Any problems in downloading this? I think it's very straightforward, so you just have to click it. So it's not downloading, or it's slow? Okay. So you must have got these templates in the Swiss model. So according to you, which template should we use? If anybody can tell that. With somebody said the one with higher GMQE score. Yes. Yes, yes, the one which is written that it has been verified using this X-ray crystallography, and as I know that this protein binds with complexes with this 2-oxoglutarate and iron, so we will take the second one for our building our 3D model. So this one, we will take this one for building the model, because it has been validated by this X-ray crystallography as well, and also it has been written that it complexes with this 2-oxoglutarate and iron. So these are the cofactors, and I know that for this protein, so that's why we will select this, and after this we can just click on build model. Yes. Uh, the purpose is just here to demonstrate you guys, and we can simply use that as well, but as I have told that in CRISPR we can get certain variations in this in our sequence, so that may be the reason that you would want to build your own model. So that's why I have just demonstrated that you have to search for the template and build the model accordingly. Otherwise, how you will get different protein structure if you just take the structure available online. I hope it's clear. So after the structure is built, so we can just download the structure in PDB format. Okay, so the most commonly used format is PDB in case of protein. So we will just click on this and it will okay. And for the next step, you will require this UCSF Chimera to be installed. So please let me know when it's installed. Okay. So everybody installed this. Okay, I can wait. Those of who have installed, they can just simply open it and have a look how it appears. And okay. Having any issue with internet, you can just copy it from somebody. I think it will work, but you have to have the same operating system. I think that's the minimum requirement. So everybody managed to get this 3D structure? Okay, so please download it as well. Yes, you have to download it in PDB format. If somehow you are not able to install it or download it, you can share it in pen drives as well, so we can wait for some time as well. Okay. Install नहीं हो रहा. So after opening this, you will get this kind of home page. You can just click on file and click on open, and after that you can open the structure you have downloaded. Like I have downloaded it and named it 16 docs. So, and after that you can click on open. Okay, so everybody has opened this now. Okay, okay, I can wait a bit more. Done? Okay. ऑप्शन कहाँ पे आ रहा है ये लिगेंड वाला? अभी तो नहीं आना चाहिए आई थिंक. मतलब आपको सिंपली यूज, मैं एक बार टेम्पलेट, अब मैं वही बता रहा हूँ, वी कैन जस्ट रिमूव एनी अदर टेम्पलेट और एनी अदर आयंस एज वेल इन दिस टूल. सो वंस यू हैव डाउनलोडेड इट, सो with the help of this you can visualize any protein of your interest, and if as you can see that there's this one red dot in the middle, so this is with the ion, so we can also remove and label them as well. So for removing this you have to go to this select, because that's not a part of the protein, that's something which was downloaded along with the protein, and we wanted the protein to be pure in this. Like in the upcoming things which I am going to tell you, we require the protein to be fully pure, like without any ligands or any ions that we don't want. So for removing anything, there are different tabs you can see, like file, select, actions, presets, tools, favorites, help. So this you can also check it on YouTube. Various tutorials are available for this as well. So for selecting, we can click on this select, and here we can select and go on to this chain section to check whether there are multiple chains available in the protein or not. So there is only a single chain, that is A, and if you click on that, everything is highlighted with this green font, and you can see that red ball is not, or because it's an ion, so it's not highlighted because it's not a part of the protein. That 16-dox protein we have downloaded, so we have to remove this part. So for that we have to do the opposite, like we have to go on to the chain select option first, and after that chain on, and we have to select that dash that is representing this any other thing besides our protein structure. So after clicking on that part is highlighted in green, and you can move this protein structure to see it clearly, and you can move it by pressing the left click and moving your mouse or trackpad. And after that you have to select on the actions button, which is adjacent to adjacent to it, and after that you have to click on that atoms/bonds, and from here you can just delete it, like it's in the bottom, and if you press this delete, this will disappear from your structure. So now your protein structure is free from any ions or ligands. So this is your pure protein structure for 16-docs. You can again save it. If you don't save it, okay. Okay, and you have to save it as well. If you don't save it, and the next time you will open it, it will again come. So you have to save this particular protein as well. For this you have to click on file, and you can click on save PDB, okay, from the drop down menu, and after clicking on save PDB you can save this and name it accordingly, okay, and you can save it wherever you want to save it. So besides this, have you all saved? Okay, so besides this we can also take we can also use this tool for because I think you must have selected the whole protein as well. So I think this bit is clear, that how we can remove anything which we don't require instead of our protein. So besides this, we also can change the models, like the interactive models. that is present in presets, like we can change it to appear something like colorful, more colorful like this, and we can also change it to appear like this. Okay, so we can also have this confirmation like this. So these are all the different models. So I think the first one is good, or we can just use the preset one that appears by default. So after that, you can also select particular amino acid residues, like if you want to highlight all the alanines, arginines, or any other amino acids present in this structure, you can just select, for example, you can select histidine, any residue that you want to highlight, and after that you can go to the actions tab, and you can go to the color, and you can select any color for that particular residue. And if we select like this magenta color, so all the histidine residues will be marked in this with this magenta color. So like if you want to, how this can be useful, like if you want to see the I think you all must be aware that we have active amino acid residues in protein. So this can be helpful in highlighting those active amino acid residues. Like those active amino acid residues fold in such a way that they comes close together in a close affinity, so that they can interact with their ligand. Like this is how this works, the active amino acid residues. So you can highlight different amino acid residues and highlight them, and which can give you a rough idea that where your binding pocket will be formed. Yes, please. Okay, वही select पे जाना है. Okay, select पे जाके residues वाले को select किया, तो आप यहाँ से कोई भी सिलेक्ट कर सकते हो रेजिड्यू, लाइक एलेनिन, आर्जिनिन, ल्यूसीन, है ना. उस पर क्लिक किया तो वो सारे सिलेक्ट हो जाएंगे, एंड वंस इट हैज बीन सिलेक्टेड यू कैन जस्ट गो ऑन टू दैट एक्शंस टैब वेयर यू कैन go to the color section, and then you can select any color which you want it to be highlighted, and after clicking on that you will get this bit. So are you finding it useful, like, or not? If we talk about protein, if you are targeting something, then you can also target your gene accordingly. Like you know that one active amino acid residue lies in exon 1, and one lies in suppose exon 2, and the other lies in exon 3, and they all come in close vicinity when they are folded in a protein structure, and they form a triad, active amino acids triad, and at that region, suppose say that is responsible for binding with 2-oxoglutarate in this case, as I have taken 16 docs. So if you have targeted different, these all three exons, like exon 1, exon 2, exon 3, which forms the active amino acid residues. So with this you can be more sure that whether the protein is formed, then it will not be functional protein, or it will not bind to say like 2-oxoglutarate. That is the compound for it. Okay, so I hope that I have made it clear that why we are doing it like this. So okay, so this tool UCSF Chimera also has some extensions as well. Like when you will click on the tool section, you will see that there are various tools for analysis. Like you can also do protein ligand interaction or docking, for which you will need the extension for Autodoc Vena, and you can do it with any other extension as well. But This supports autodoc Vina quite well. Have you ever heard about this autodoc Vina? Okay. So in docking, what we do that we provide the protein and its substrate or ligand, whatever we can call it, and we provide artificial conditions like in silico, so that they can bind if they are compatible. If they are not compatible, they will not bind. So for that reason, I was telling you that there are certain active amino acid residues that can also be predicted. Like there's a whole lot of in silico thing that is involved before doing experiments in wet lab. So there are a lot of things as well. So for have you all cleared that what is docking? Basically, we want to study that whether the substrate binds with our protein or not. And if our protein is somehow edited, it will not be able to bind with our protein. Okay. So for that, we will require an extension for Autodoc Vina. And if you want to learn further about it, there's free tutorials available on YouTube, and various combination of these tools can be used. Like one other tool for protein visualization is PyMol. You can search it and download it if you want to, otherwise I can just open it for you. Like several tools have their own advantages and disadvantages. Okay, so this is another tool for protein visualization that is most commonly used besides UCSF Chimera, and it's called PyMol. So this is how it looks, and the various tutorials are available for all of these tools. So here's the functioning is pretty much like same, like we can click on file and open our molecule. Okay, if you don't want to download it, you can just have a look. So okay, so this is our structure, 16-doc structure. Okay, here we also have the option of displaying the amino acid sequence. Like when we go on to this display tab, and we can click on sequence, and this will show us our amino acid sequence associated with this protein. So various tools have their own advantages. Like in this PyMol, we can select any particular amino acid and replace it with any other amino acid. So that will create an artificial mutant for conducting our in silico experiments. Like it has also highlighted the amino acid that I have clicked. Like I have clicked this, it is also highlighted here, and if I replaced it with any other amino acid, it will create an artificial mutant. So with this we can also say that with what kind of mutation our activity of this protein will reduce or up regulate. So before proceeding to do wet lab experiments, I think this is very good. I'm not sure whether you are getting my point or not, because otherwise you have to perform all the laborious experiments and you are not sure that whether this will work or not. But you have if you have done the same in silico, using in silico approaches, then you are very much sure and you have more probability of getting the same result. So that's why these in silico studies are important. Like it's important to know what are the advantages and what are the importance of using these in silico tools. So that's why I am emphasizing more on these. So coming back to UCSF. Okay, so for this protein, I know that the substrate for it is 2-oxoglutarate. So from where we can download these substrates? Like, do you know that we can also build these substrates as well? Like there are various tools where we can add particular amino acids one by one, and we can build our substrates as well. So but let's not go into that. But if you want certain compounds, you can just go on this pubchem. You can search pubchem and it will open this home page where you can get any substrate for docking purpose. I'm talking about docking purposes. So you can just open this pubchem and you can write 2-oxoglutarate or the short form for it. Let me type the full 2-oxoglutarate. Okay. Okay, so the first compound is 2-oxoglutarate. It also has various other names. So we can just click on it. Right, and this is how it looks in 2 dimension, like with this model, and we can download its 3D structure from here at the right corner with this download tab. You can also download the 2D structure as well, but we want 3D structure, so we'll download this. And here we cannot download the PDB structure, as it only give us in SDF format and JSON format, XML format. These are all different formats, so we will here download it in SDF format. So we will save it, and I have previously saved it, so I won't download it again. So let me just open our molecule once again, because we have labeled it, so I'll be opening it again so that it appears good. Okay, so this is our 16-dox protein. So now we will open this 2-oxoglutarate also. So for that we'll simply insert it. Okay. Okay, you will simply type the name of your substrate. Okay, so after downloading, so here I am opening this substrate, that is 2-oxoglutarate. You can open more than one protein at a time as well, if you are able to understand what's it's appearing in the screen. So here this is our compound 2-oxoglutarate that is colored. This I think. Please let me know when you have opened it simultaneously with your protein. Okay, opened. I think I have taken a long time, so let me just wrap up. Okay, when you have opened both the proteins and the substrate and its substrate as well, then you can do the docking, for which you will require the extension for Autodock Vina. So okay, let me just tell you in a download after downloading it, or okay, You have already clicked on it. So okay. When you have introduced both the protein and its substrate, then you have to go onto the tools where you can select go on this drop down menu, and you can stay on this surface binding analysis, and there you can select Autodoc Vina. But this will not work in your case because you haven't downloaded the extension. Okay, so you have to download the extension as well. So it works on it will work, but you have to download it first. So I think I don't have much time so that you can download it, but you can you have easily access to the tutorials that are freely available on YouTube. So after that, this box will appear. So here the first option is the output file, where you can type the name of the output file that you want. Like we can simply write output here, or any other name you want to give, or we can just type 16-dox underscore 2OG, that is 2-oxoglutarate. Okay, 2OG, and after that we have to just provide our receptor and ligand. Like in the receptor box you can see that our 16-dox is already there, and in the ligand we have to select that 2OG because we have named the molecule 2OG. So it's with this drop down we can select our substrate. So after selecting that, the next step is to build a grid, and that grid is basically coordinates like in all the 3 dimensions x, y, z, and it provides the exact let me just show you first. Okay, you can either enter the coordinates yourself, like manually, or if you know where your active triad or your active amino acid residues lies, so you can select it using this, like using your mouse. So like say it's your active amino acid residues are somewhere in this region, so you have to select that region where you are sure that your substrate will bind with that protein. So like I have randomly selected using this and made this grid. Like now you know what grid is, like the area of the protein that will be covered for binding, where it will look for any potential interactions. So after that, when you will build this grid, you must be seeing that there are several values which appeared here, like 0.6, minus 7.7. This all depends on the size of your grid. Okay, so after selecting this, we have several options. Like for docking, we generally we can add hydrogens or remove them as well. If we want to add, we will click on true, and if we want to remove them, we will select on false. Similarly, we have to we can also remove non-polar hydrogens as well. You can check for the tutorial for this, and you can do it yourself as well. Okay, so after this, we can also go to this advanced option where you can select the number of binding models. Like it provides us models, like different positions where the substrate can bind. Like if we have selected it to the max, that is 10, so that it will give us 10 models for our substrate to bind with the protein. Okay, so the higher it is considered to be good. So we can also tweak it, like we can select it for 1 and a maximum value of 10. So the exhaustiveness is this, the higher it is, the better it is. Okay, you can read about it as well in the information of this tool. So with this, if you have successfully done these steps correctly, like you have to just provide the software with the receptor and the ligand, then you can just click on OK, and it will produce an output file which we have named here. So this will give you an output file, and from there you can analyze it as well. So I think that's all from my side today, but you can read about it more and